Happy Friday, everyone. My name is Kristen Spencer, and I'm a medical oncologist at the Rutgers Cancer Institute of New Jersey, where I specialize in treating patients with biliary tract cancers. Welcome to this edition of the Cholangiocarcinoma Foundation Translational Webinar Series. It is my distinct pleasure to introduce you to Dr. Paula Olaizola from the Biodonostia Institute of Spain. And she will be speaking to you on targeting NAE1-mediated protein hyper-NEDDylation and cholangiocarcinogenesis. This is meant to be an interactive session, so if you have questions, please feel free to ask them or send them in the chat. So without further ado, Dr. Olaizola.
Hello, good morning, good afternoon, everyone. Thank you to the organizers. Thank you, Kristen, for the nice introduction. And yeah, I'll start talking about my work. As you said, it's about targeting NAE1-mediated protein NEDDylation, halting cholangiocarcinogenesis. So cholangiocarcinoma comprises an heterogeneous group of malignancies that mainly affect the bile duct epithelial cells, also known as cholangiocytes. Although its epipathogenesis remains elusive, its incidence is increasing worldwide, currently being the second most prevalent primary liver tumor after hepatocellular carcinoma. And the geographical distribution of cholangiocarcinomas is asymmetrical, being its incidence higher in Southeast Asian countries. And such discrepancy, it's likely due to the increased exposure to the specific risk factors, such as, well, particularly the liver fluke parasites. So cholangiocarcinomas are generally classified according to their anatomical location in intrahepatic, periular, and distal cholangiocarcinomas. Traditionally, periular and distal cholangiocarcinomas have been referred to as extrahepatic cholangiocarcinomas. So intrahepatic tumors arise from any portion of the intrahepatic biliary tree, whereas periular and distal cholangiocarcinomas can rise from the hepatic duct or common bile duct, respectively. So the different subtypes of tumors present differences in epipathogenesis, risk factors, incidence, and prognosis. Cholangiocarcinomas are generally asymptomatic in early stages, being commonly diagnosed in late phases when the disease is already disseminated. These, together with the high chemoresistance of these tumors, highly compromises the therapeutic options available, mainly based on surgery. Therefore, patients with cholangiocarcinoma present bad prognosis and survival rates below 6 months after diagnosis. For all these reasons, it is very important to understand and unveil the molecular mechanisms involved in cholangiocarcinoma pathogenesis in order to look for new therapeutic targets. So in this regard, different genetic alterations have been described. However, only around 40% of them are druggable. In addition, epigenetics have been shown to play an important role in the initiation and progression of these tumors, altering tumor phenotype in the absence of changes in the DNA sequence. Nevertheless, the mechanisms underlying cholangiocarcinoma pathogenesis beyond gene expression have not been explored yet. So this is where we focus on posttranslational modifications, which refer to the covalent attachment or proteolytic cleavage of a functional group or a protein to a substrate protein. These are chemical changes that can alter the structure and properties of individual proteins, affecting their stability, activity, localization, and/or interaction with other molecules. To date, several post-translational modifications, or PTMs, have been described, for instance, ubiquitination, methylation, phosphorylation, and their alteration has been observed in different diseases because they participate in multiple cellular processes and are now being targeted with promising results. However, in this work, we will focus on protein NEDDylation, which refers to the covalent binding of NED8 to a lysine residue on a substrate protein. NED8 binding to a protein occurs through an enzymatic cascade that involves 3 steps: activation, conjugation, and ligation. First, the NED8 precursor is processed by a NED8 specific protease, and then through an ATP-dependent manner, NED8 becomes activated and can bind to the heterodimeric NED8 activating enzyme, which is formed by 2 subunits, NAE1 and UBA3. Next, NED8 is transferred into one of the conjugating enzymes, UB2F or UB2M, and finally, a substrate-specific E3 ligase can bind the NED8 to its target. The NEDDylation process is dynamic and reversible because there are deNEDDylating enzymes that are able to remove NADH from its target. So what are the substrates of NEDDylation? So cholings are the best characterized substrates, and cholings are a molecular scaffold that, together with an adapter protein, a substrate receptor, and a ring protein, form the family of choline ring ligases, which are well-known ubiquitin ligases. So once cholings become NEDDylated, choline ring ligases are activated and they can ubiquitinate and target for proteasomal degradation different substrates. Among the choline ring ligase substrates, we can find important mediators of cellular processes such as cell cycle, apoptosis, or senescence. In addition to cholings, other substrates have been reported to become NEDDylated too, and these include transcription factors or kinases that are also involved in important cellular processes such as proliferation or apoptosis. So NEDDylation, protein NEDDylation, is modulating or is involved in multiple biological and cellular processes, and therefore its deregulation has been associated with disease. Indeed, different hepatic disorders have been reported to exhibit aberrant protein NEDDylation. And to date, the most common strategy to modulate this pathway is by targeting the activation step with Pevonedistat, a first-in-class inhibitor of the NADH activating enzyme, NAE. So, taking all this information, we hypothesize that aberrant protein NEDDylation may contribute to the development and progression of cholangiocarcinoma, representing a potential target for therapy with Pevonedistat. So first, we aim to analyze the expression of the NEDDylation components in human cholangiocarcinoma. To do so, we first evaluated the transcriptional levels of the regulatory subunits of the NADH activating enzyme, NAE1, in cholangiocarcinoma tissue samples from 5 independent international cohorts compared to controls consisting of normal bile ducts and surrounding liver. As it can be observed, NAE1 expression was found upregulated in cholangiocarcinoma tissues compared to controls. Importantly, NAE1 expression was upregulated independently of the underlying mutation driving the tumor, as it can be observed when we stratified the patients according to the most common mutations in cholangiocarcinoma. Furthermore, NAE1 upregulation was particularly observed in moderate to poorly differentiated tumors and in tumor epithelia compared to tumor-stroma. Similarly, yes?
Some question for you here on that slide. Did you look at upregulation of NEDDylation machinery and other underlying mutation types such as HER2 or FGFR2, and if not, why?
So that's a very good question, but no, we didn't do it because the data we had, it was basically on these 3 mutations and the rest were organized in the Indeed, yes, we should check for other mutations like FGFR2 fusions and stuff that could be different. Yeah.
Great, thank you.
Okay. So similarly, we looked at the expression, we analyzed the expression of the NEDDylation pathway ligand, NED8, in cholangiocarcinoma tissues and found it was also upregulated in the 5 cohorts studied. So then we moved on to looking at the NAE1 and NAD8 protein levels at different stages of the disease. So we were able to see that NAE1 and NAD8 were almost absent in surrounding liver tissue, whereas the staining was very strong in pre-malignant, pre-invasive lesions and grade 1 and grade 3 intrahepatic cholangiocarcinomas. And this could suggest that NAE1 and NAD8 overexpression seems to be like a common phenomenon in cholangiocarcinoma, and that can already be detected in pre-malignant stages of the disease. Then we moved on to cell cultures, and we again analyzed the expression of the regulatory subunit of the NAD8 activating enzyme, NAE1, and the ligand NAD8 in 5 cholangiocarcinoma cell lines compared to normal human cholangiocytes. So similar to what we observed in the biopsies, NAE1 was found upregulated in all 5 cholangiocarcinoma cell lines compared to NACs, whereas the ligand NAD8 was only upregulated in one of the cell lines. Then we aim to evaluate the protein NEDDylation levels, like the NAD8 conjugated proteins, and by immunoblot we were able to observe that NAD8 conjugated proteins were elevated, were more, we found more NEDDylation of proteins in cholangiocarcinoma cell lines compared to normal human cholangiocytes. So once we identified like the NEDDylation machinery was being overactivated in cholangiocarcinomas, we next aim to identify which proteins, which NEDDylation targets were involved in cholangiocarcinoma progression. To do so, we performed an immunoprecipitation of NAD8 conjugated proteins, and then we did a high throughput proteomic analysis of those NEDDylated proteins, comparing normal human cholangiocyte versus cholangiocarcinoma cells. And we found around 240 to 250 proteins, from which 69 were differentially expressed between cholangiocarcinoma cells and normal human cholangiocytes. 12 were found downregulated and 57 were found upregulated in cholangiocarcinoma cells. And the biological function of this protein was analyzed by gene ontology, finding out that these proteins, the upregulated proteins, or the upregulated NEDDylated proteins in cholangiocarcinoma, were mostly involved in processes such as protein stabilization, protein modification, cell survival, cell cycle, and cell proliferation. So seeing that these kind of processes, of biological processes, were being associated with increased NEDDylation in cholangiocarcinoma, we next aim to evaluate the effect of Pevonedistat, a first-in-class inhibitor of the NEDDylation pathway, in the pathogenesis of cholangiocarcinoma.
Paula, perhaps you can, before we move on to the pharmacologic inhibition,
yes,
maybe ask some questions about, have a little discussion about some of the data you've presented so far. Yeah. This is probably a better time to do it than when I interrupted you previously.
No worries.
One of the things I was curious about is, did you see any variable expression of the NEDDylation machinery by anatomic subtype across the cohorts?
So we did look a bit into that, and we apparently saw no significant differences between intrahepatic, perialar, or distal cholangiocarcinomas. However, we did this kind of analysis in different cohorts, and I do remember there was one which might have shown that I think it was extrahepatic cholangiocarcinomas might have less expression of the NEDDylation pathway components. However, the different cohorts we are analyzing, they have a different number of patients there. So maybe if we could reach a better number, have more patients to analyze, the data would be a bit more like, you know, I wouldn't say trustful, but like
a little more robust.
Yeah, more robust, exactly. That's it, thank you.
Great.
So yes, so apparently there might not be differences between tumor subtypes, but it's something like we should study in detail, like with more patients.
I see. Okay, great. And I guess I heard you mention that there seemed to be a correlation between, I believe it was NAE1 expression with differentiation, I think poor, moderately, or well-differentiated tumors. Did you see any correlation with or look at other pathologic features such as lymphovascular invasion or nodal positivity, or outcomes such as survival in these patients?
Yeah, that's interesting. And we did check, and we saw no association with, like lymph invasion or tumor recurrence, for instance. But we did see in some of the cohorts, again, we did see some association between high expression of NAE1 or high expression of NED8 and worse survival. But again, it was in some cohorts, not in all of them. So it's something that we should further study, like to get robust results on this. like increase the number of patients in the study, and we will see like really what's going on.
Great, thank you.
Okay. So yeah, then we went on to see what was the effect of Pevonedistat, like the selective inhibitor of the NAE1 pathway. So first of all, what we did was to confirm that indeed Pevonedistat was able to inhibit protein NAE1. So after incubation of normal human cholangiocytes as well as cholangiocarcinoma cell lines with Pevonedistat, we did see a reduction of the NAE1 levels of proteins. So we did confirm its value, the value of Pevonedistat as an inhibitor of the pathway. And then we moved on to doing functional assays. So we tested the effect of Pevonedistat in proliferation, and we were able to see that Pevonedistat inhibit proliferation of both cholangiocarcinoma cells and also normal human cholangiocytes, although the effect was a lot more prominent in cholangiocarcinoma cells. Then we also studied the effect of Pevonedistat in cell cycle, and we were able to see that Pevonedistat induced cell cycle arrest at G2M. That was something that it had been previously published for other kind of tumors, and this was accompanied by an accumulation of cyclin B1, but this one wasn't active because the increased phosphorylation of CDK1. And we also observed an accumulation of the cell cycle mediators V1, P27, and P21, which are all choline ring ligase substrates. So if we're inhibiting NAE1, choline ring ligase substrates won't be degraded. So we do see like the whole NEDDylation process is being stopped. So we have in cells arrested at G2, and this is something that's regulated by the DNA damage checkpoint at this point. So this is a checkpoint that prevents mitosis initiation in the presence of damaged DNA or incompletely repaired DNA. So we wanted to know whether Pevonedistat was able to induce some kind of replication stress or DNA damage in these cells that could lead to the arrest at G2. To do so, we evaluated the abundance of ORC1 and CDT1, which are two choline ring ligase substrates again, and that are involved in DNA replication. And after incubation with Pevonedistat, we saw they accumulated. And their accumulation could be triggering some kind of replication stress that could be activating this DNA damage checkpoint and therefore arresting the cells. Indeed, afterwards, we did by immunoblotting and immunofluorescence, we observed an increase of the DNA damage marker phosphorylated histone 2AX, which confirmed that Pevonedistat was able also to induce some kind of DNA damage. So the DNA damage, the cells arrested at G2, made us think that Pevonedistat could be inducing apoptosis of cholangiocarcinoma cells. And indeed, by flow cytometry assays, we were able to see that Pevonedistat induced cell death in cholangiocarcinoma cells. And this was also reported by the increase of apoptotic markers. So Pevonedistat induces cell death, but also has other kind of effects. So here we see three-dimensional spheroids of cholangiocarcinoma cells, and Pevonedistat led to the shrinkage of these spheroids. And additionally, Pevonedistat impeded or halted colony formation of cholangiocarcinoma cells. On the other hand, we all know that the combination of gemcitabine and cisplatin is the main treatment, the first-line treatment for cholangiocarcinoma. But we tried a triple combination with Pevonedistat, and we saw that Pevonedistat sensitized cholangiocarcinoma cells to this treatment, which was very interesting. And the combination of gemcitabine and cisplatin can sometimes be only palliative because of the appearance or acquisition of chemoresistance, because cisplatin has been described to generate single-strand DNA breaks, which can be repaired over time. So we wanted to know what kind of DNA damage was Pevonedistat inducing. And to do so, we plated cholangiocarcinoma cells and we treated them either with cisplatin, Pevonedistat, or the combination of both. And after 12 hours of treatment, we did see an increase of DNA damage marker phosphorylated H2AX in all conditions. However, we left the cells for another 12 hours of recovery with normal media, without compound, and we saw a drastic decrease of this DNA damage marker in the cisplatin condition. And this suggested or showed that cisplatin-induced DNA damage could be repaired, partially repaired at least with time, whereas the DNA damage induced by Pevonedistat wasn't able to be repaired. It was stable. So next, what we did was evaluate the effect of Pevonedistat in vivo models of cholangiocarcinoma in order to see whether the therapeutic strategy of inhibiting NEDDylation could be a good therapy for cholangiocarcinoma. So we first evaluated the effect of Pevonedistat in a subcutaneous model of cholangiocarcinoma, where cholangiocarcinoma cell lines were subcutaneously injected in the posterior flanks of immunodeficient mice. These mice were then homogeneously distributed into two groups, either vehicle-treated or Pevonedistat-treated groups, and the tumor growth was monitored over time. So as you can see, Pevonedistat was able to halt cholangiocarcinoma, subcutaneous growth of cholangiocarcinoma cells, and this was due to an increase of apoptosis, as we can see by the marker caspase-3. We then also evaluated the effect of Pevonedistat in an orthotopic model of cholangiocarcinoma, where luciferase-expressing cholangiocarcinoma cells were directly injected in the livers of immunodeficient mice. And then mice were randomized into two homogeneous groups according to the luciferase activity and treated either with a vehicle control or Pevonedistat. So interestingly, vehicle-treated tumors grew over time significantly, whereas the tumors treated with Pevonedistat show no significant growth between the starting point and the time of sacrifice. And this is a bit of the lesions we can see in the livers with hematoxylinosin stains. So next we aim to determine the, Kristen.
Oh, no, go ahead. I was just
okay, sorry. Yeah.
I was just going to ask a couple questions here too. I think it's another good time to stop.
Yes.
For those of us that perhaps aren't as versed in the lab language that might be attending right now, could you explain a little bit about what spheroids are and why you chose to incorporate them in your experiment?
Okay, great. Yes. So spheroids is a type of cell culture that it's in 3D basically. So instead of plating the cells in flasks and we have a 2D culture, now we can do this kind of three-dimensional structures that are more similar to what we can have maybe in humans in vivo, because they are not attached to anything. They're more interacting with each other and we can see a bit more. So it's a really interesting model to see the effect of in this case, Pevonedistat, but all the compounds, to see whether the three-dimensional structure is working the same as the 2D culture, and it's more representative, let's say. It's okay?
Great. Yeah, that's great. That's very helpful. I noticed that one of the highlights of treatment with Pevonedistat is that it causes unrepairable DNA damage. Do you think that sort of negates the role of combination therapy?
No, that's a good question, but I think the combinations should be still there. I think it might potentiate or sensitize more to other compounds too. Like, I think still it's important to combine Pevonedistat with other kind of drugs, for instance, also like targeted therapy, for example, or immunotherapy, or even like other kind of cell cycle inhibitors. It's something to continue to analyze and continue to study, because I think monotherapy, it's going to be very difficult. So I guess always trying to combine different aspects, also seeing the molecular profile of the tumors in order to be more directed to it. Yeah, I think something like that.
Do you have any sense at all at this point what the mechanisms of escape might be with this kind of treatment, or is it just too early to know that?
That's a really good question. I honestly I don't really know, but it's something like we should study on, and like we should move forward to doing those things, because and to what I know, there's nothing really described on that, and it'll be very interesting to find what's going on.
Yeah, for the future. I don't know how much clinical exposure you've had with the Pevonedistat, but I do know that this is seemed to cause a little bit of a milder cell death in control cells. So are you aware at all, is the toxicity what has been expected, or has it been more toxic than we thought because of its effect on normal control cells?
So Pevonedistat has already been approved by the FDA for myelodysplastic syndrome, and different clinical trials have been trying different dosages. So it has been shown to have some toxicity at high doses, but the ones we are working on here, they're supposed to be below that toxicity. And indeed, in the in vivo models, we saw no problems with the mice. But yes, it's something like in the clinical trials, it should be further studied. And the effects we see in normal human cholangiocytes could be maybe improved if we adjusted the doses. I'm not really sure on that, but yes, we are working on doses below toxicity ranges, like quite below. So I guess like once the toxicity is set, like if we work below that, it'll be fine. Again, like the clinical trials are working, like there are different clinical trials ongoing with this, and they'll provide like more answers to these questions.
Great, absolutely. I do see there are questions in the chat, I think, or in the Q&A. So I think let's just keep moving, and we can pause, but we'll definitely have a discussion at the end where we can get to some of the future prospects and things that people are asking about.
Okay.
Thanks.
So yeah, next we aim to determine the impact of molecular targeting of the NAE1 pathway in the pathogenesis of cholangiocarcinoma, and to do so, yes, we used the CRISPR-Cas methodology to generate an NAE knockdown. Briefly, we designed the guides that then were cloned into Cas9 expressing plasmids, and these plasmids were then transfected into cholangiocarcinoma cells, which were then selected by sorting through GFP selection. So these cells were then expanded and tested. So we were able to get different clones, but we show only one in here. It had reduced levels of NAE1 expression through qPCR, also reduced protein levels of NAE, and this led to reduced protein methylation levels in total. And in order to see what was the different proteomic profile between the NAE1 knockdown cells and the control cells, we performed proteomic studies, and we identified around 5,000 proteins, from which 781, I think, were differentially expressed. 409 were found downregulated, and 372 were found upregulated in the NAE1 knockdown cells. And it was interesting to find that the downregulated proteins in the NAE1 knockdown cells were associated to biological processes such as cell cycle, DNA replication, DNA repair, or protein localization. However, the upregulated proteins were mostly related to energy metabolism or drug response. So similar to what we observed with Pevonedistat, NAE1 knockdown cells presented reduced proliferation and an arrest in the cell cycle too, with accumulation of the P27 and P21 cell cycle mediators. Again, NAE1 knockdown cells showed an impaired capacity to form colonies and were unable to form the three-dimensional spheroids. And that was probably because they presented a reduction of stemness markers such as CD133, OCT4, or SOX2. Later, we aim to determine the impact of NADylation impairment in cholangiocarcinoma cells on their subcutaneous growth, tumorigenicity, sorry. So then control cholangiocarcinoma cells and NAE1 knockdown cholangiocarcinoma cells were subcutaneously injected in immunodeficient mice, and tumor growth was monitored over time, and it became evident that NAE1 knockdown cholangiocarcinoma cells presented impaired growth. Moreover, we performed an oncogene-driven cholangiocarcinoma model in NAE1 heterozygous mice compared to wild-type mice. These NAE1 heterozygous mice and wild-type mice were subjected to hydrodynamic tailwind injection with the AKT and NICD1 plasmids together with a sleeping beauty transposase. And after 3 weeks, mice were sacrificed and livers were examined. So as you can see, the wild-type livers presented pre-neoplastic lesions, whereas the NAE1 heterozygous mice presented minimal or no lesions at all. And indeed, the volume of pre-neoplastic lesions in the wild-type animals was significantly higher. So finally, we aim to ascertain the role of NADylation in the crosstalk between cholangiocarcinoma cells and the tumor microenvironment. Cholangiocarcinomas are very desmoplastic tumors, and this highly compromises the outcome of patients and response to treatment. Cholangiocarcinoma microenvironment is composed of tumor-associated macrophages, cancer-associated fibroblasts, neutrophils, infiltrating lymphocytes, as well as extracellular matrix. And this all supports the proliferation and growth of the tumor. So in this work, we particularly focused on the role of NADylation in the interaction between transformed cholangiocytes and cancer-associated fibroblasts. To do so, we first isolated cancer-associated fibroblasts from 2 cholangiocarcinoma patients that went on to surgery. And after isolation, we characterized them with CAF markers such as platelet-derived growth factor receptor beta, fibroblast-activated protein 1, alpha-smooth actin, or vimentin, and they showed no expression for the biliary epithelial marker CK19. So once we had isolated and characterized these CAFs, we moved on to defining what kind of role they were having on cholangiocarcinoma cell growth or proliferation or migration. So we saw that CAF-derived media was able to induce proliferation, spheroid growth, as well as migration of cholangiocarcinoma cells. And then we did something similar with the supernatant of CAFs onto the wild-type and NAE knockdown cholangiocarcinoma cells. And we saw that CAF-derived media was able to induce proliferation and migration on both control and NAE1 knockdown cholangiocarcinoma cells, although the effect was less in the NAE1 knockdown cells. What we did later was the other way around. We took the supernatant from control and NAE knockdown cholangiocarcinoma cells, and we added it onto CAFs to see their effect on proliferation and migration. Interestingly now, we saw that the supernatant, the conditioned media from the NAE1 knockdown cells, reduced the proliferation and migration of the CAFs compared to the control-derived media. So seeing all these differences, we decided to study what was going on in the supernatant, like what was the secretome of these cells, of the control and NAE knockdown cells. And we analyzed the secretome. We found like 600 proteins, from which 213 were found differentially expressed between the secretome of the NAE1 knockdown cells and the control cholangiocarcinoma cells. The downregulated proteins were mostly associated with fibroblast activation, fibroblast migration, angiogenesis, but also protumorigenic and profibrotic pathways. So this was very interesting. And then we also did a small trial on Pevonedistat, using Pevonedistat to test the viability of CAFs. And we saw that Pevonedistat was able to induce some kind of death and reduce the viability of CAFs, which was important too. So in summary, in baseline conditions, cholangiocarcinomas present an upregulation of the NADylation pathway, which leads to the degradation of ring lysate substrates, as well as to the degradation of cell cycle mediators, which lead altogether to the proliferation and growth of these cells. They also secrete some factors that induce the proliferation and migration of CAFs, which in turn can also promote the proliferation and survival of cholangiocarcinoma cells. However, when we treat the cholangiocarcinoma cells with Pevonedistat, we inhibit the NAE1 pathway, and this leads to DNA damage, replication stress, cell cycle arrest, and proliferation inhibition. Also, Pevonedistat can directly affect CAFs, and the inhibited cell can have some kind of negative interaction with CAFs, reducing their proliferation and migration. So finally, as a conclusion, we can say that aberrant protein NAE1 contributes to cholangiocarcinogenesis by promoting cell survival and proliferation. Targeting protein NAE1 reduces cell proliferation and tumor growth in experimental models of cholangiocarcinoma. Abnormal protein NAE1 impacts on the cholangiocarcinoma stroma crosstalk, and inhibition of the NAE1 pathway with Pevonedistat may represent a potential therapeutic strategy for patients with cholangiocarcinoma. So finally, I'd like to thank the Liver Diseases Group at the Biodonostia Health Research Institute, as well as all the international and national collaborators, as well as to the organizing entities. Thank you.
Thank you so much. I mean, this is obviously a fascinating area, and I think something that is really sorely needed to look at in many tumor types, particularly cholangiocarcinoma. I think, you know, most people are really probably thinking about questions regarding combinations. And before we get to those questions, though, I'm wondering, was there anything in this project that surprised you at all?
So, yes, I mean, in the beginning, sometimes like you start using compounds and you don't know really if it's working or not. But in this case, Pevonedistat was really easy to work with. Like, it was very easy to see results quickly. Also, something that was difficult, let's say, was the CRISPR, because we weren't able to get a complete knockout, and that was at the beginning it was a bit frustrating, you know, because you'd like to have the knockout, but we did see like animals, the complete knockout of animals went viable. And we thought like, okay, the NAE1 pathway must be important, because if we try to remove the whole thing, the cells aren't living. So that was interesting too. That was something like good. And then when moving on to the crosstalk between the tumor and the stroma, like finding like some proteins like that could be modulating that, I think it's very interesting. Then that's something like we should study more, because we're focusing on the cells, on the tumor cells, but like the tumors, the whole thing, like CAFs are supporting the growth of the tumor, and tumors are at the same time supporting the growth of the CAFs. And again, going into studying the macrophages and the matrix and all of that would be very interesting, because you can see there's an interplay that's important.
Right. And there's a question in the chat that first compliments you on your wonderful presentation, and then does ask about this very topic about why you focused on crosstalk between the cholangiocells and CAFs. Is the NEDDylation pathway also important for other cell types found in the tumor microenvironment, such as the TAMs?
So thank you very much. We focus particularly on, so we first moved on to studying the tumor microenvironment, because we did see that the NEDDylation impaired cells, like the NAE1 knockdown, had a reduced cell secretion. We did see some differences there in the proteomics. And we knew like cholangiocarcinomas are very desmoplastic. They depend a lot on their environment. So we decided to study CAFs. But what we've seen is like probably studying macrophages, studying endothelial cells will also be very important in this case. And we haven't really studied if NEDDylation is affecting macrophages or endothelial cells, that we've seen, like, even if it was a small experiment, but Pevonedistat targeted or could reduce the viability of CAFs. So it would be interesting to see that in all the cell types too. It's probably deregulated, and I think there are some studies already going that way.
Okay. So one of the questions is asking about, is there any experience so far combining anti-NED with anti-PD-1 immunotherapy?
So that's very interesting. I think there's nothing in liver, but I remember there was something in glioblastoma, for example, like using immunotherapy and Pevonedistat, and showing that Pevonedistat was able to, again, like make the effect of the immunotherapy like better. And again, so yes, I think testing those kind of combinations is what we should do now, like try to improve, because in the end, I think what patients will need, it's combination of different therapies in the end to improve.
Right, and I know, here, even just from the data you showed today, it seems like combinations, I mean, really, there's no end to combinations. I'm thinking of, whether there'll be synergy with other cell cycle inhibitors that can sort of target other parts of cell cycle progression, since we know that cells are often in different stages going through the cell cycle at different times. Including, I believe you had some preclinical data that showed that there's involvement of non-homologous end joining, and I'm wondering if things like DNA PK inhibitors might synergize as well.
Yeah, you're absolutely right. Like, we should try every combination we can on this, like see what's the best combination to arrest this, to stop this. But yes, it's a bit of a never-ending story, maybe. Like, there are so many combinations we can try, but I think that's the way we should focus the work, like the future work, on like trying to do combinations and see what's the best for each cell type.
Right. And I'm wondering, the CRISPR results that you saw, and sort of the mechanism of resistance or delayed or differential response you were seeing there, do you expect that to also occur in vivo, such that something like an autophagy inhibitor might be helpful, or not really?
So that's something we should try, definitely. It could happen in vivo, and again, yes, maybe some kind of autophagy inhibitor or some other kind of inhibitor could boost or could stop that chemo resistance, but it's something like to consider for future studies. Thank you.
Yeah. So what do you see? We've asked a lot of questions about combinations and things of that nature, and your answer may be the same here, but what do you see the future of this research being? Where do you see it going?
So yeah, on the one hand, of course, trying immunotherapy on like Pevonedistat and immunotherapy, Pevonedistat and other kind of inhibitors. I think also moving on to the tumor microenvironment studies, like it would be important to see like what's going on. It's not only the tumor cell, but the ones surrounding them. And that's something we can also do in vitro. For instance, the 3D spheroids, we can make them like multi-cell type, let's say, like put in the CAFs, the cholangiocytes, and maybe also monocyte, macrophages, whatever, and try to see if the effect's still the same or if there's something improving or even going worse, like what could we do there? And maybe also trying to do combination therapies between Pevonedistat and targeted therapy in cells that we do know which mutation they have, and there are targeted therapies for that one. So I think that will be very interesting to transfer into clinics for the personalized medicine aspect of it.
Right, it would especially be interesting if as you move on to sort of some of the targetable underlying mutated tumors, the FGFR2s and HER2s, that would be
exactly. Yes.
Yeah. We have another comment in the chat, again, complimenting you. It's a fascinating subject. I think we all agree about that. And sort of still under the theme of future research, can you say anything about anti-NED with other cancers, particularly GI cancers?
I do know they've been doing trying anti-NEDDylation therapies in different cancers. I'm sorry, but I do know more about liver. I know there's been studies in hepatocellular carcinoma and also in fibrosis, which could be, but I think it seems to be like a common phenomenon in all tumors, because glioma, breast cancer, I think it's also, and it's something to take into account. I honestly don't know especially about GI, but I think it could be happening. In the end, NEDDylation occurs in all cells, and it's involved in multiple cellular processes. So probably the cancer, like the tumor cells, is taking advantage of it, and it's a fast way of making the cell do stuff. Like it's a fast way of activating or inactivating proteins, and the tumor cell probably needs this fast mechanisms in order to survive. So it's probably happening in there too, but we'll have to further look at that.
Great. Well, I'm not seeing any other questions in the chat, so I just have one more for you, Paula, and please feel free if you have any I can answer, that would be helpful. I'm just wondering what's next for you. What's your next project?
So we are working also in this kind of story, but with stimulation, which is another post-translational modification. And there's also been like selective or specific inhibitor. So we're trying to do kind of the same story and to see if at some point we can converge both stories to see if, again, combination. So yes, something like that, further studying on the tumor microenvironment and seeing what we like to do on this.
Absolutely. I think this is a really, really exciting area. So I look forward to following your work and hopefully bumping into each other in person one day at one of these conferences.
Yeah.
So yeah, I'd like to thank everyone for attending, and without further questions, I'd like to thank Dr. Olaizola for joining us and the Cholangiocarcinoma Foundation for making sure that this translational seminar series is an ongoing success. And I hope to see everybody at the next session. Thank you.
Thank you.